we are back, and we were talking about examples of where cells in the body use active transport as a means of taking in excessive amounts of something, or releasing excessive amounts of something against that substance's concentration gradient. And so we looked at action potential or nerve impulses sent through neurons for communication within your body, which is absolutely essential for everything that you do. We looked at how plants, root hair cells, are adapted to absorb minerals and water even when the concentration inside those cells is greater than the soil in which it needs to use active transport to absorb those minerals. And now we're looking at sugar absorption in the gut, or the absorption of any substance that needs to be pumped against its concentration gradient. So if you were to look inside a human, inside their intestines, you would see that the intestinal wall is kind of furry looking, which is a little bit unappealing. And this is down to these finger-like projections that look like this. And on these flappy projections, you'll see even smaller flappy projections, and these are called villi, or villus plural. Villi? Villi plural. And if you zoom up even closer on these villi here, to a singular villus, you will see that the epithelial cells of this villus have tiny villi of their own, called microvilli. So you're increasing the surface area inside the gut hundreds and hundreds of times by adding all of these folds, folds upon folds upon folds. And that's because the whole job of the intestine is to absorb water and nutrients. Here we go. So basically, in any animal, sugar or any nutrient has to move into the cells of the intestine. So when we were on that previous slide, these cells being the epithelial or surface cells, really close up of the intestinal wall. So those cells have to suck up all the sugar, and they send that sugar through capillaries into the bloodstream where it can be transported around the body to do everything it needs to do. So in short instances, those epithelial cells are gonna absorb enough sugar to be at high concentration relative to outside. But we need to keep bringing in more and more and more sugar, because that's eventually going to be sent out into the bloodstream and onto the rest of the body. So if the body was only using facilitated diffusion or protein carriers to get these molecules across, then once they reached equilibrium, they would start moving back the other way. And you would never really get to a point at which the epithelial cells were absorbing enough sugar that they could actually sustain the entire body, because they would never get to a point at which they had more sugar than the inside of the gut. We need to be able to absorb as much sugar out of that digested food as possible, even if the cells at the very surface of the gut are, for a short instance, at high concentration. So these epithelial cells, again, this is the inside of the gut here where all the food is, and this is the epithelial cells, and you can imagine a bunch of other cells here because the intestinal wall is not one cell thick, but eventually it reaches the bloodstream, and at that point glucose can diffuse through a protein carrier, because it is still a very large molecule and can't just slip through the membrane itself, but it can diffuse into the bloodstream. However, at this end over here, the inside of the gut, at first after you have a massive meal, there's a lot of sugar here, and it's not such a consideration as to how you're going to get that sugar into the cells. But after a meal, when your gut is at low concentration of sugar and you've got some residual sugar in the cells, how are those cells going to accept more sugar in if they're already at a relatively high concentration? Although they want to, they're going to need to pump that glucose against its gradient into the cell so that it can keep processing the sugar into the body. And in this case, it's going to have to use protein pumps. So movement of a substance against its concentration gradient. And because this takes up a lot of effort, it is going to require the pump to use up ATP, otherwise known as cellular energy in the process. Move. Cool. So these are our 4 things that we've covered so far. We've looked at passive transport from high to low concentration across a membrane, simple diffusion just in between the phospholipids, facilitated diffusion using protein channels and carriers, and now, and osmosis as well, which is simply this form of diffusion, but in particular the diffusion of water. Now we're looking at active transport. The very first process of active transport being the use of these protein pumps in the membrane, going from low concentration, looking at these orange squares, to high concentration here. And yeah, next we're going to be looking at the second mechanism behind active transport, which isn't part of this diagram, called cytosis. So we've looked at pumps. Now we're looking at cytosis, which is that cell eating kind of thing. So cyto, I always like to look at the etymology of biology words because they always make a lot of sense and they make them, in my opinion, a lot easier to remember. So cyto, we're usually talking about a vessel or a container. So cytoplasm, for example, is a container of jelly, plasm being, a jelly substance. And cis is often a suffix we have on words that are to do with a process taking place or an action. So cytosis being the action of a vessel, and that's essentially what it is. It's another way of getting substances into a cell, and it's used when we want to get a really large quantity or really large pieces of a substance into the cell that can't just be pulled through pumps or carriers or anything like that. We can break cytosis into 2 forms. So the cell taking things in, endocytosis, so endo meaning inside, and the cell pushing things out, or exocytosis. So again, it's this vessel that we're considering and a large quantity of a substance, but whether it's going in or out, we can use the words endo and exocytosis. It's really quite simple how it works. Basically, the plasma membrane, which surrounds the entire cell, that phospholipid bilayer, pinches in and eventually fuses around, a large quantity of a substance or a large piece of something until it forms a totally individual vesicle or phagosome and transports this substance wherever it needs to be in the cell. Because it takes energy for the phospholipids to do this, we consider it active transport. So it has to use up ATP in order to do this. So we've said endo and exocytosis are the 2 branches of cytosis, being bringing things in or pushing things out. Within the bringing things in process, endocytosis, we can separate that even further into bringing things in that are liquid and bringing things in that are solid. So consuming something solid is called phagocytosis, and consuming something liquid is called penocytosis. I don't know how this helps me because it's stupid, but it might help you. I think pina colada, even though it's not pina colada. Drink, pina colada, drinking, penocytosis. If that helps even one of you, then I'm less embarrassed. Just reading. So, an example of this. We looked at examples of the other form of active transport. This is a really, really big use of phagocytosis in the body, and it is by the aptly named phagocytes, or a type of white blood cell whose entire job is to go and eat naughty things in your blood. Okay, so they find, using receptors on their membranes, they find things that seem foreign to them. And why is this GIF not working? It's a good one. Oh well. Maybe it'll work in the slides. Yep, it basically eats or drinks quantities of something that are foreign and then uses its lysosomes, which are these kind of bubbles of enzymes, to dissolve those particles and then spit them out again if they're harmless. Okay, here's a real electron microscope photograph of a white blood cell. That's this yellow stained thing here, engulfing anthrax, which is a really dangerous bacteria that you do not want. So I think this is one whole anthrax bacilli here. Whee! So yeah, that's pretty amazing. And once it's in there, it'll use all of its enzymes to digest and dissolve that naughty bacteria and then spit it out somewhere. Oh, fun fact, When these phagocytes are done, they basically implode and become pus. Yummy. So when you've got bacteria in your skin, all of your white blood cells will go there and they'll destroy the bacteria, and then they kind of just form a big white ball, and that's your acne. Hope you enjoyed that. So videos here that you can watch. This is an awesome one by Crash Course. Again, it covers, I think, all of the active transport and passive transport processes. There's a cool poster here that you can look at that, again, puts all of these things next to each other, comparing and contrasting. Keywords from this lesson that we have talked about: protein pumps, okay, pumping something against its concentration gradient, such as the sodium potassium pump in your neurons. The use of cellular energy, or ATP, being really what sets apart active transport from passive transport. Cytosis being broken down into bringing things in, endocytosis, pushing things out, exocytosis. Endocytosis being split into phagocytosis, or eating, penocytosis, pina colada, drinking. Whether this forms a bubble of a liquid, we would call that a vesicle. Oops, sorry. And whether it forms a bubble containing a solid, such as a bacteria or something else naughty, this would be a phagosome. And a phagocyte is a white blood cell whose whole job is to do this process, and it uses lysosomes to do so, which are full of enzymes. So those are some keywords that you should be able to confidently use. Again, recapping everything that you should be able to do at the end of this lesson as kind of a whole topic so far recap, because we need to move on. And that's it. So I'm going to be setting up some Education Perfect and requesting some SciPad pages. I'll put those also up on Facebook, Google Classroom. So you have heaps and heaps and heaps of stuff to do. I would definitely recommend going back and making thorough notes on this stuff so that you understand it when we get back, because it is massively important for this external, which I think is 4 credits. Correct me if I'm wrong. Okay, bye.